Yeah, first of all, I congratulate the KIET School of Pharmacy for organizing this kind of an excellent workshop in association with the AICTE. And I'd like to thank Director and Principal and Professor Vaishali Patel for this opportunity to address and to share about our research work in the particular domain. So let me start with like kind of is right now we this very important subject on how this computational biology or chemistry or like the virtual it's enabling the drug design. Now we are witnessing this kind of advancement of the current pandemic. The computational chemistry or this virtual screening really made a lot of advancement in the drug discovery process. Right now, some of the drugs, including vaccine designs, are originated from this basis of the computational chemistry, biology, or this informatics or virtual screening. Is a kind of a subject of integrated one. Like you need to have chemistry, you need to have mathematics, you need to have some physics, and also finally applied in the biological systems. So with that, like this talk, since this is the first talk in the workshop, so I'd like to have some broad coverage of how this computational chemistry and biology in drug design, some of the fundamental concepts, or like kind of what practically we are looking at it, and how we are doing at CSIR-IDR in our group. Like we are developing some kind of a key models for alternative to safety and toxicity assessment, and how we are trying to understand the molecular level interactions by studying host guest interactions. And finally, I'd like to highlight, if time permits, like some of the highlights and on our computational toxicology efforts and database development. So CSIR-IITR is one of the pioneer research institute in the toxicological research, and we are working all niche area of toxicology like system toxicology, health risks assessment, and food drug chemical toxicology, regulatory toxicology, environmental toxicology, advanced material, nanomaterial toxicology. Everything culminated into translational research. Recently, we have established computational predictive toxicology is an area called toxicoinformatics, and also we have established some high performance computing facility to strengthen these research activities. So our research activities are very pretty much very well defined. Like our idea is to develop a alternative platform for replacing this in vivo and in vitro methods. So we have to develop a platform of innovative predictive approaches to address toxicological issues. Like for that, we have to do like fundamental understanding and predicting toxicity of the biological system, how these contaminants, chemicals, drugs, they are interacting with the biological system like DNA or proteins or several other targets, how they are interacting using various level of approach like quantum mechanics, molecular dynamics simulation, or further system biological approaches. And further, we started developing some kind of a robust platform called chemoinformatics, but that is also integrated with translational bioinformatics. So that will provide you like robust screening, like kind of very fast and rapid way we can screen chemicals. And we also establishing several databases relevant to toxicology. And also we provide services to industry in the particular like kind of active pharma ingredients or their toxicity and developing some other new inhibitors. These are all the things we are very closely associated and working with some of the industries, including people involved in drug design. So this is one slide I'd like to highlight here, like how the computational methodologies to extract molecular level informations to expand, or like how that can be utilized in our various biological and chemical applications. So if you consider the computational methodology in a core domain that has like we call them as a quantum mechanics methods. That's what's the principles of Schrodinger equation. So that one you solve that particular atom include in inertials. So those will be counted in this Schrodinger equation, and that will solve the particular representative of the particular atom and the representative of the compound, and will get the properties of the particular chemical system. So this one is give you much more accurate level of information on the properties and chemical reactivities like bond forming, bond breaking, and how they interact with each other. But the disadvantage of this is like limited to number of atoms and like limited to like smaller systems. So then to overcome this, the molecular dynamic simulation or molecular mechanics is developed that's based on Newton's law of motion. When the two atoms connected with the bond will connect the angle, then we develop a force field. That force field represent the huge system that can be utilized like that function of time, how the molecular behave, like how this drug go and interact with particular target and how it's stabilizing or destabilizing the system. That one can study using molecular mechanics, molecular dynamics simulations. So there is in between, we started working on quantum mechanics and molecular mechanics, where the reaction or interaction happening, we can treat them in the higher level quantum mechanics and rest of them are molecular mechanics. That way, the computational tools are expanding its limits to address several issues. So it's kind of replacing various experiments, what currently people are working at the bench side. So our goal is to go and how this virtual capability, not only in molecular medicine and drug design, it's also working on several different domains. I think you are various diverse field. People are coming and attending this interview, attending this workshop. Like to, these are the few important areas. Like this is only a list. Like there are n number of application and domains that virtual capability can expedite the realizable solution and actionable insights in these sectors like molecular medicine, personalized medicine, preventive medicine, gene therapy, drug development, or kind of a domain where there are n number of activities one can do using computational approaches. So we also currently working on some waste management, cleanup, and alternative energy resources, or several other biotechnological problems also. Currently, we are using our in CSIR idea. So one of my project I working with closely in ICMR Indo-German product or antibiotic resistance. That's also like that's relevant to our drug designing capability as well as developing a new inhibitor to overcome this critical challenge. That's really become very big problem in this AMR and multi-drug resistance. So how the computational understanding of the overcoming this efflux pumps of particular pathogens, so that will give you the lot of boost for drug developing process. And rest of them are also like very important, like crop improvement, insect resistance, and drought resistant variety development, and how to understand the allergenicity and veterinary medicine, veterinary science, and food science. Also currently in our lab we are working on it. These are the highlighted in red. We are currently working in our lab. Drug development is integrated process for us, like to predict chemical toxicity. That's where how we connected with this drug development process. So the key points of this talk is to, as I said earlier in the outline, to provide a concept as well as a practical introduction to the methods and tools in computational drug discovery, and to understand the capability and limitation. That's really one need to understand where one can apply and what system you can choose for your study and how reliable they are and how different methods give you an advantage over studying the systems. So this is very key point, but I'm going to touch upon very briefly on this topic, and also going to give you some more integrated approach currently we developed in our laboratory on chemical, including drug safety. So when you see the drug development process is a kind of a pipeline, like that includes several segments, like identifying targets and developing, choosing some key lead molecules and characterize them and identifying pathway. And for us, like in computational toxicology, we work mostly on the preclinical and clinical domain. So that's we call them as a regulatory toxicology. Then finally, it will go to in the marketplace. How then we can go for whatever the where screening that can taken into preclinical and clinical, and that can be translated. But it's kind of more very like tedious process and also very critical process as well. So when we looked at the data of clinical trials conducted between like the year 2000 to 2020, around more than 3 lakhs and more than 3 lakhs, 3.5 lakhs as on July 14. And after that, there is huge hike due to our COVID-19 pandemic. So the most of the drugs in the clinical trial is fails due to this toxicity issues. So that's in order to overcome that, we have to study not only toxicology, we have to entire chain of drug discovery process one can perform using computational approaches that will having expedite your drug designing process as well as understanding of how one can develop some of the key molecules as a suitable drug or inhibitor. So, what is computational-based drug discovery? I think your School of Pharmacy, the team members and resource persons, many of you can definitely give you a very, very fundamental and basic basic fundamental understanding and give you a great lecture on that. Dr. Vaishali Patel is the expert in this area. I think they also are working on a lot of computational approaches. Just I was looking at that research like outcome, and it's really great to share some of the overlapping concepts, like how we can providing some kind of unique insights using our computational efforts. So computational based drug discovery, what we can predict, how we can analyze, how efficiently we can translate them. These are the key questions like when we started doing this kind of work. So when you looked at the pipeline, as I said earlier, so you definitely need a multidisciplinary approach, not only like single domain based interest. And that's what I was like trying to cover from the beginning. There are not only computational approach, also there are a lot of integrated approaches out there. As I said, quantum mechanics, molecular dynamics simulation, virtual screening, and so many things can be integrated together. Right now, the system biology approaches are greatly involved in the adverse outcome pathway analysis. That enable from end to end expertise in computational toxicology or computational drug designing process. Sorry, the toxicology always is very close to our heart. So whenever it comes computational means like it's a computational drug design in this talk. So this pipeline include several components that's called like if you find out one target disease, if you want to have a disease like tuberculosis or any other diseases like for example our COVID-19. So you need to identify some of the targets, like if the COVID-19 proteins, probably many of you guys aware of, many of or all of you aware of, like COVID-19 has a structural protein and non-structural protein on the membrane. So we target them to develop some of the inhibitors or drug to stop its biological process like replication or transformation and whatever, so on and so forth. So that gives you some of the approaches in the bioinformatics or cheminformatics or drug designing process, like target identification and genomics. And you can use like various bioinformatics tools like proteomics and system biology approaches to develop the particular targets. And then followed by, you have to do a chemical information approach like bioinformatics and cheminformatics, like how one can choose some kind of lead molecule or analogs for the lead molecule if it's found to be some of the disadvantage or you want to fine tune some of them. So that will work on some of the screening tools like virtual screening, like docking, molecular docking, autodock, and so on and so forth. One can do and identify the screening of the particular library of compounds against the target. So once you did this process, you can identify a drug which is potent and more like kind of suitable for in terms of binding, in terms of stability, in terms of several other factors, like what are the physicochemical factors you are counting upon. That will give you an lead molecules. That lead molecules you can optimize or develop some kind of further analogs, and you can go for drug refinement process. And also you can do ADMED properties or like adverse outcome pathway. Finally, you can go for like kind of very, very selected sets for your experimental approaches. So the target identification is like we have several targets, like receptor you can choose, enzyme you can choose, gene target you can choose, hormone or ion channels. So that is the first part, like how you start your drug discovery process by identifying your direct molecular targets. That fundamental concept is that the targets are a cellular or molecular structure involved in the particular pathogenesis or the disease conditions. Okay, that's your target. And then going for a cheminformatics process, there are few other methods are available. Here I would like to highlight only on quantitative structure activity relationship and pharmacophore model. I think some of the other lectures they are going to cover in detail. And this quantitative structure activity relationship is like if you know one structure, the similar structures also having a similar functions. Okay. Once again, sorry. Sir, hello. सर ये टॉक चल रहा है सर ये ऑनलाइन टॉक चल रहा है सर वही तो ओह सर 1:30 one just now yes sir just now I think started sir. Yes sir my KIET sir KIET yeah yes sir thanks sorry for the disturbance and actually yeah one of the urgent meeting they are calling but still it's fine and I'll finish it by 1:30. So the QSIR is one of the very important approach. Like if you know the compound which has similar structure, the other compound which is resembles to the same structure of your identifiable compound, that's also having similar functions. The property of the structures which are similar, their properties also pretty much work in a similar manner. That's what the thumb rule for quantitative structure activity relationship. So in silico models are very highly effective and that's and kind of a rapid and high throughput process. You can screen n number of compounds, like you can also screen millions of compounds, lakhs of compounds for identity. That is the advantage of using this particular QSAR models. Like when you looked at the domain, how it's going and interacting, if you started doing molecular dynamic simulation or quantum mechanics methods, that will also require some time. So this one, the QSAR models, like if you have already pre-built databases and data sets or rules, you can immediately screen a larger data sets and come up with the structures which are having that kind of a property you're looking for to develop a drug or any other suitable compounds for your research interest. So in silico models provide high throughput process needed to handle a large volume of impurities. Like here, we just focus on some of the impurities when we are developing a API, like active pharma ingredients. So we are looking for the impurities. Like I'll touch upon some of the key concepts in impurities, what we currently using for ICH M7 guidelines. And that will give you a fundamental rules and kind of a process for selecting a compound which are much more safe in terms of effective and safety. And the QSIR model now it's kind of various European agencies and other FDA and all like they are recommending for utilizing before starting your experiment, particularly on mutagenicity test, like putting active compound in active pharma chemical impurity identification. This is not the QSIR is not that kind of a one recently emerged like what we are currently calling machine learning or artificial intelligence. Those evolved in the period of 19 when you started looking at your atomic table. That's where our QSIR started, structure activity relationship. So it's a kind of series of process. Now it's in the stage where we can use advanced machine learning and artificial intelligence tool to get a kind of a like much more sophisticated algorithms that will give you better correlation and better relationship patterns. So as I said, it's identify correlation between chemical structural features and biological activity. So one can have like if you understand the process of QSAR models, then you can start at building around the what kind of models that will be suitable for your predictions. So here the two models we call them as a statistically derived model and expert based model. Like that's kind of a part of QSAR approaches. So just skip this. So this is kind of one of the overflow of workflow of QSAR models. Like when we started doing a QSAR, building a model, you first started collecting some experimental value. In our computational toxicological QSAR models, we started collecting our EC50, IC50, LC50, or some other properties of toxicological endpoints, which are experimentally determined. And also, if you want to say some other physical chemical properties also, you can collect them in order to validate and develop a relationship of those models. So once you collect the data sets, we started collecting a series of compounds. That's based on that only you collected the data set. Those series of compound structure will be evolved in various tools and also the techniques like quantum mechanics method, molecular mechanics method. Now so much virtual screening tools are available. So using that, you develop a descriptor, you develop the properties, physical chemical properties like um, ionization potential, electron affinity, or number of heterogeneous, number of atoms present in the system, or so many things, so many descriptors of physical chemistry, physical descriptors, topological descriptors, graphical descriptors, so many descriptors are available. You started predicting those, calculating or computing those descriptors. Once you have the experimental value, you have the descriptors like the predicted calculated properties, you started understanding them, how they are related to each other. There are statistical methods like linear regression analysis, multiple linear regression analysis, partial least square methods, artificial neural network. As I said right now, other artificial intelligence methods, those methods are enabling the statistical methods is much more stronger and superior to develop models. Once you have developed you can do some two cases like test set and training set. So you can do that training set and start predicting the model which has highest correlation. You can do, like, for example, y is equal to mx plus is a linear regression equation. If you got some really good correlation coefficient value, take the model and take the training set and build the model and start predicting your test case. Once you are seeing like much more satisfactory correlation and prediction with your reference data as well as the predicted data, go ahead and that your model is built on in QSAR development. For example, we are developing a QSAR quantitative structure toxicity model. This is how we develop some toxicity models of series of compounds. Once I have the model, we started predicting for the new compounds. So that's the advantage of this robust QSAR based to modeling. So this right now, that's what I said you is like kind of a preliminary process, like kind of a fundamental process. Now we are having your database based research tools. Like if you have a rules called experts-based rule-based model and statistical derived models, that's already having a pre-exist database. Like if you just give your structure, like for similar to some kind of a pharmaco-based model, if you have a similar structure, you just go and see the database is having that particular alert in the system. That's called expert rule-based models, like human written rules, data analyzed manually. That will give you a qualitative prediction and broad range of endpoint covered. So if you have a particular group present in your target chemical compound, if you go and apply this experts-based model in the database, so that will provide you some qualitative prediction of various properties you are looking for, like inhibitions or like toxicity. So for us, like broad range of toxicity endpoints are available in this, some of our whatever the computational tools we are using. Using that, we kind of narrow down our searching process or understanding the particular compound much more detail, whether that will go for how to take it the compound further or like how to understand its toxicity. The statistical derived model also is another QSAR models. Currently, these are all much more advanced tools. We are currently using it, like hierarchical model built using machine learning techniques. It's fully automated. If you know the structure, that will provide a quantitative prediction. And also various mutagenicity endpoints also we can predict using this statistical derived model system. As I said, expert-based tools, like if you have this kind of a structure for us, like because this more mainly we are focusing on toxicophore based approaches. So as I said, it's a pharmacophore based models also, like pretty much that's what we are trying to cover it here. So the matching part of the query of the particular structure, if it's found, then that will give you an alert. Statistical based model is like kind of a data sets available based on your query that will build a network and that will give you a decision tree and that will provide your data for your prediction. So what we do based on this expert and tool based analysis, we follow a ICH M7 guidelines. So we develop different classes based on our obtained properties, and that will go and decide whether the molecule, what should go with the experimental testing. Whether if it's a known mutagen, no carcinogen, that's it. Then no, mutagen with unknown potential for the carcinogen, then we can go for some of the other testing condition. Is proven non-mutagen, then you can start doing that molecule for uh, another level of testing, like clinical testing or preclinical testing. So mutagen with known potential or carcinogen with known potential, then we'll do some kind of bacterial assay or some kind of a in vitro methods. Then we can test them whether they are toxic or non-toxic. Then we can go ahead with the further processing. So that's where the part of chemoinformatics all covered. So another one important is ADMET properties like adsorption, distribution, metabolism, excretion, and toxicity. So there are several endpoints are available, and based on how this oral availability, how it's going to be distributed, what are the targets, it's the key important points like it can cause blood-brain barrier or not. And currently we are developing some blood-brain barrier based new models because many of the models are sometimes like it's available online. They are really good and validated with certain systems, but it's not for entire series of compounds. So we are started developing our own models for this kind of ADMET properties and as well as toxicity endpoints. So once you are having a satisfied of carried out all this entire process, that will give you an handful of system. Then you can go add further, you can analyze that adverse outcome pathway. So that's where the toxicological part is very, very important right now. So it can go and search the drug space, how it's going to interact with the protein in network, and what are the metabolites it can be formed, and how we can go and the biotransformative pathways and identifying the targets it's going to bind. So these are the things one can understand using this pathway analysis. So I'll like skip this pathways, like kind of if you see here, we call them as a components of node, pathway, and network. That node symbolizes a list, or for example, it's a gene that especially is one-dimensional representation of data. Pathway is how the nodes are interconnected with each other. That essentially is a two-dimensional representation of the data. A network is the entire collection of cellular functions and the regulation involved in the pathways, particular pathways. So currently we are using this for example, some of the chemical compounds. Some of the we develop an QSIR based approach, identifying their toxicity endpoints or structure based understanding. Finally, we are taking them into some kind of expression of this data and how they are going to interact in this biological pathways, and how it's what are the targets it's going to impact upon and what are the consequences of those target interactions. So traditionally, it's a kind of a tedious process, like do some bioassay, biological lead optimization, and preclinical clinical studies, and finally validated targets. And current target-based discovery include like target identification, like high throughput testing system and target hit. So the system biology approaches further, it's reducing this process of drug discovery. That's what I would like to suggest, like highlight here. this system biology based adverse outcome pathway analysis also is very important of integrating into the drug discovery pipeline. There are n number of software and tools for drug designing are available. Like as I mentioned, for each component, like databases, docking, ADMAD, binding site prediction, draw tools, molecular modeling tools, and homology modeling tools. So many of you, or many of them are like open source, and some of them are like commercially available. So you can use whatever the way you want. Currently, many of you you can't go to lab, or sometimes they cannot go to office or work. So these computational virtual screening tools are giving handy, and also create your scientific curiosity how one can utilize the system. And actually, I congratulate all of you, all the participants, for showing this interest and having a huge success for this workshop. So there are several databases also available. Currently, what we do like zinc databases, which has the already pre-existing compounds are available. Once you identify the lead molecule, you can order them and started doing testing. That's what we are doing for our antimicrobial resistant AMR of inhibitor designing projects. And again, this is the some of the pathways analysis we are using, particularly Pathway Studios and BioPanther, the Panther one. And there are several pathway analysis tools also available. So now I go into, these are the fundamental concepts and kind of various components of drug discovery pipeline using virtual approaches. So now I'm going to show you some of our R&D work and how we are utilizing these integration approaches for relevant chemical toxicity or drug safety predictions. So I don't need further highlight upon this, but still I'd like to show you yeah, why computational chemistry and biology tools are very, very important here. So there are more than 60 million chemical compounds or exist. But in 2011, that time itself, and every month or every year, more than 4,000 new chemical compounds are adding into the repository. But very, very limited data sets are available on long-term and short-term toxicity in the National Toxicological Portal. That's also require huge amount of money as well as time. So if you want to do entire data sets, it's really, really a like kind of a Herculean task. So the computational tools are become really handy, and these are all as providing an alternative instead of in vivo, in vitro, in silico provide an advanced edge to expedite this drug discovery process or safety information. So as I said earlier, more than 80,000 chemicals are registered and more than 1 million compounds, we call them as a high production volume chemicals. These are produced in every year, but only 43% of SP chemicals have no testing data on the basis of toxicity. So definitely we have to create various toxicology information like systemic toxicology and organ specific toxicology if you consider what kind of toxicology information you want from us. Like if you compare this experimentally, there are many toxicological endpoints like as I listed here, acute toxicity, subchronic toxicity, or chronic various toxicity endpoints, or hepatotoxicity, neurotoxicity, reproductive toxicity. There are many endpoints that involve many mechanisms. It's very, very tough problem. Okay. So computationally also is very, you need to be very focused when you start working on developing various computational understanding and drug design of from the beginning of target identification, interactions, and further refinement. That's what we call them is absorption, distribution, metabolism, and toxicity properties. And another important thing is like we definitely need alternatives, like not only for the pain, distress, or like using of animals, but how they are accurate. So there are the information provided by the US National Cancer Institute. There are 5 to 25% accurate of the rodent model, and in vitro methods are based on the precise screening and all, like that's all 80 to 80% accurate. If you integrate with computational tools with proven some in vitro, in vivo methods, definitely you can generate one of the next generation risk assessment for these drugs and chemicals that will give you much more sophistication in the reliability of your prediction and reliability of development process. So recently, this kind of concepts are currently evolving with integrating various computational and omics and new in vivo, in vitro methods that will give you a next generation risk assessment tools. Look, first we start with in silico approaches like identifying the scenario and identify the molecular structure and collect the existing data and do a clear screening of computational chemistry based approaches. Then you can go further based on you are refining the process. That definitely enabling this risk assessment of next generation process much more sophisticated and much more targeted rather going on all sort of testing process. So this is one of the very important thing currently we are trying to work and integrate with our experimental groups in CSIR idea. To reiterate, the computational process of testing is a low cost and high throughput process. It's also provide a mechanistic understanding, like how it's going to interact, what are the target sites, which are the residues it's going to interact, and what are the transformation it's happening. These are the mechanistic understanding or mechanism involved in this process. Not only this mechanistic understanding, this also provide and two, like if the particular group is doing some kind of a adverse effect, you can redesign them. That's we call them safe by design approaches. So you re-engineer and repurpose the particular group or particular moiety that will give you an edge over designing this process much more faster manner. And also further reduce the in vitro and animal testing for various lead prioritization, identifying the chemicals and drugs. So this is kind of a process, and some of the chapters also we have reported, and I can share those details in the, and you can go ahead and read further on these methodologies. So this is the traditional drug discovery process, and advanced integrated tools. This made this drug discovery process is much more faster and sophisticated for going from bedside to bedside. Okay. So yes, another thing is always any method. If you say there are a lot of advantages as well as pros and cons involved in it. Like if you say in vitro model, in vivo model, like just focus on in silico models. So advantages provide a mechanistic understanding and rapid screening, and I also identifying potential adverse effect and understanding physiology. And also right now we are with the artificial intelligence, machine learning models, we can also generate some virtual patients based on the data. So whether it's going to work or not, this is the advantage of virtual models. And disadvantage also there, like if you're not choosing the particular right group or system or the mechanism is not entirely clear, the model have some errorness. So need to be very, very careful about what are the models you are choosing, that models are relevant. Otherwise, there will be garbage in, garbage out. So you need to be very, very critical in choosing the model system. That's what is very important. And you need to do a benchmarking and you need to do a validation. So that's how one can develop a more reliable model for your using computational approaches. There are toxicological, that's what is listed here, the TopCat, hazard case, EcoSAR, Oasis, and drug SARA nexus. These are the common tools we are used for toxicity prediction. And Vega models are currently we have started using it. That's also an open source platform one can test. Many of you are aware of pharmacophore model. Here it's a kind of a toxicophore concept based on structural alert. So there are so many endpoints. Like for example, we just focusing on few of the mutagenicity and carcinogenicity properties. That's where you can first line of screening we started using our mutagenicity based predictions. So if you see this kind of common known molecules which are having this particular chemical groups, they are very well known mutagen. So once you same kind of a might is available in other systems, definitely that will going to be a similar manner. So this will provide a great advantage for us to see whether the molecules, this kind of commodities are present and screen them based on the earlier observations. So the health concerns of chemicals include skin allergen, irritants, and also cause respiratory damages, neurological impact, and systemic various toxicologies. So we thought of developing several models, like we can also develop a skin sensitivity uh, predictive model, and some of the respiratory predictive models also we have started develop, and neurological models also we developed, and systematic models also we are currently working on developing it. So for testing the chemicals or APIs, we thought of using a genotoxicity as one of the endpoints for us. Like genotoxicity is potential to interact with genetic materials and cause some measurable DNA alteration. And mutagenicity is all fundamentally the potential to change the genetic properties causing heritable and genetic mutation. So this one, conventionally, people can do some kind of a standard test like chromosome aberration, micronuclear formation, or like Comet assay. And recently, this REACH and various European agencies, they are suggesting first to start doing with in silico approaches like QSAR-based models. Then that will provide an genotoxicity-based prediction. Then you can go ahead and take this particular molecule, whether which are cleared from this QSAR-based predictions. So when we looked at the literature, particularly what are the carcinogenicity QSIR models are available or mutagenicity models are available. Particularly, we started using a Vega is a kind of open source platform for various QSIR based toxicity prediction or various other property prediction models. There are 4 carcinogenicity models and 5 mutagenicity models. So we don't know which one has more accurate kind of a system what we are going to work on. So we asked the question like to predict the carcinogenicity and mutagenicity of various specialty chemicals, and using this models like open source models. We have selected 131 chemicals from RIFAM and EDTOX database and started using these models, but these models are doing different haphazard results. So then, as mentioned to you, there is pros and cons. Here we started developing a benchmarking or validating a system. So for that reason, we collected around 2000 chemicals which are having a experimental data of rat carcinogenicity and mutagenicity from DS-TOX, the carcinogenic potency database, and IASS TOX database. So we collected these databases around 2000 compounds and started applying these models what we have chosen for mutagenicity as well as carcinogenicity. Here, as I said, now we have more and more highly sophisticated algorithm for statistical performance is available. So we can have a sensitivity, specificity, accuracy, negative prediction value, positive prediction value based on the model which are available as already available. That day, actually, as I showed you how to build the QSAR models, those models are, for example, it's a kind of a simple working equations. This is the important things like if you develop new QSIR models or utilizing the same models with the structure of your interest. So you take the series of compounds and started applying the existing models and see how they are started predicting the value of the sets which are you gave an input for that model to see the performance of those models. So here we started using the five-fold analysis. The five-fold analysis is a kind of statistical validation methods like true positive, true negative, false positive, and false negatives. So how they are relevant to each other like sensitivity, specificity, accuracy. These are the 5 model plot we made it and how each model performed in terms of these 5 selected criterias. There are 10 criterias also available. We started using this also for some other cases. So in this particular work, we use the five-fold analysis, cross validation analysis. Furtherly, we got the ROC curve that identified which model is performed better for the 2000 chemical compounds what we have chosen. Then we using this model, we started predicting for this 131 compounds, which is our interest specialty chemical, which are we are very much interested in. In that also what we know, like some of them experimentally available data we have collected, and started comparing with our prediction as well. And we also made a new analysis called overlapping analysis, among which are overlapping between carcinogen and non-carcinogen, or mutagen with carcinogen, or carcinogen with non-mutagen. This kind of overlapping analysis we have performed. And finally, we got a selected set of compounds which are no, and that also we further validated the experimental data. So we developed a model which is much more robust and accurate are with the prediction of this particular mutagenicity and carcinogenicity using existing tools, existing machine learning models. And the second part of this work, what we started doing is like there are a lot of things emerging, fields are emerging currently, like additive manufacturing. Additive manufacturing is nothing but 3D printing, right? People have started using 3D printing for developing various prototypes or um, models, or like sometimes real products also developed using 3D printing approaches. And bio 3D printing also is kind of getting more and more popular and still evolving for our toxicity testing. That's also we are working on developing some kind of organ models using bio 3D printing. So this work, when we started, many of the polymers, whatever used for 3D printing in materials, are plastics, like aceto, nitrile, butyl, butadiene, and stearins. And some of the polylactic acid or bioprinting also available. That is very limited applications. Depends on application. People started using different fibers. Those fibers releasing the chemicals, right? So that's where we thought, okay, what are the chemicals is going to be released, on how one can develop a risk assessment platform using computational approaches for characterizing this chemicals and fibers used in 3D printing. As I said, it's a kind of market is really growing, and various fused filament materials are utilized in the system, but that's also coming with the another risk. So we need a risk assessment framework, pretty much like your drug designing, right? Drug screening process. So when we looked at this particular issue, we saw this polylactic acid and ABS filaments. It has a high emission potential of this particular ABS. So we thought of creating a smaller data sets. What are the components of this particular ABS fiber? So we found like around 40 chemicals are released, and we collected those chemicals and started predicting various key parameters like absorption, metabolism, digestion, excretion, and other toxicity endpoints like QSIR based endpoints and express based endpoints like inhalation toxicity, oral toxicity, carcinogenicity. And this is some of them are open tools like National Toxicological Portal, also available various open tools for predicting this toxicity. And express based system also, because when they tested this emission of the real patient, real like people working in this industry of 3D printing, not 3D printing, it's like people in UK, I think they conducted a study when people are exposed these kind of factories where they are making these fibers. They found like acute toxicity of many of them, like oral or some kind of respiratory tract infection. But long term, also people need to consider for it. That's why we started like kind of a systematic analysis of using this particular ABS and the chemicals released from ABS at the higher temperature. So among which it will give you, when you utilize these computation approaches of these models, that will clearly categorize into what are the chemicals falling highly toxic and extremely toxic or non-toxic. And also you can see how when you are using this same chemicals, what are the how they perform in terms of various in vivo model system like rat and mouse. This is also computational models are available, and some of the models are really good and you can use them. And we used this rat inhalational LC50 values and other from National Toxicological Portal, and that gives a category of the compounds which are going to impact in terms of toxicity. And we also use exposure-based tools that will give you probable, possible, and equivocal level of grouping of various toxicity endpoints. That also gives us insight into how this molecule behave, how these compounds coming from this 3D printing behave. So then we further started predicting absorption, distribution, and ADMET, all the properties, and how it can cross blood-brain barrier. And very interestingly, there are some of them are having higher to medium blood-brain barrier penetration that we reflected upon our um, pathway analysis. So we used the Campbell here, like identified various protein targets which are relevant to these chemicals involved in our study. We identified 194 protein targets and of chosen chemicals. These chemicals are associated with various pathways. If you say they are crossing the blood-brain barrier, we found they are affecting that particular targets. So it can cause various immunological signaling, impact them, neurological signaling, cardiovascular signaling. This will provide a complete information of how these molecules or compounds going to impact. And that will provide you, if you want to develop a new fibers or new materials, we can use this framework. Okay, we have some validated level of risk assessment framework. We can use this new material and what are the compounds are coming out, we can screen them with more reliable way. So that's how we develop this framework for 3D printing and particularly for ABS filaments, and this can be used, this framework, for further testing. So these are the whatever I talked about is kind of a robust screening. And now you can go for some specific interactions, like how this particular dye, what we are using, going to interact with DNA in a major group, DNA with minor group, or it going to intercalate. These are the studies also one can do. Here, I would like to highlight one of another translational project what I carried out like almost a decade ago. So this is Microbacterium tuberculosis cell wall. This cell wall containing a lipoarabinomannan, is a glycolipid that has a mannose cap. When it has a mannose cap, is considered to be more virulent compared to uh, lipoarabinomannan without the mannose cap. So this present in the cell wall of the tuberculosis, Mycobacterium tuberculosis. So what we envisaged in this project is to develop an yeah, testing system, like kind of a pathogenicity assessment of this Mycobacterium tuberculosis, like a diagnosis of this infection, bacterial infection. And further, we see how it can do this pathogenicity. These are the two questions we thought of asking. Want to develop a diagnosis system, as well as we need to understand this process. And for the experimental group, they want to know how this molecule like lipoarabinomannan can be captured in a system which has having higher affinity to it. So we computationally, what we started doing it, we selected two membrane lipids, DOPC and POPC, which has small changes in their tails. Then we build the model for the lipoarabinomannan. There are no such like 3D models are available. So we started constructing each domain, like capping domain, core, mannose core, albino core, how they anchor the fatty acid chains. So everything got constructed using computational approaches. So once we got the model and we tested with some of the atomic, some of the other experimental tools, they told them like it's never reported. One of the reviewers told that, hey, this model looks like if you make this six of lipoarabinomannan, pretty much the huge space, whatever we observed in their experiments. So this gives us a lot of confidence to us. Like it really is a gigantic model what we built for lipoarabinomannan. We started interacting with this membrane lipids, how it's orient and how it's going to be penetrated inside the cell wall, and what are the forces responsible like hydrogen bonding interaction, or these are the non-bonding interactions, how they involved in the penetration of this particular virulent factor inside the cell. That's we have studied. And interestingly, we developed this platform of high affinity platform to capture this virulent factor that now lead into universal bacterial diagnostic platform, biosensor based platform as a sandwich assay. Now it's under recently got like R&D 100 award in United States. So that's how the computational tools enabling yeah, not only the mechanism and also developing diagnostic tools as well as the drugs. Yeah. These are some of the databases currently we are working on, and this focus database also we have created is like food additive databases. So that contains entire properties from the structure to biological impact and toxicity information and what are the targets it's going to involve and how one can develop a new additives or new chemical safe compounds for food industries. And ToxDB2 is a polyphenol toxicity database. And antimicrobial surface agents also we have developed, and mycotoxin databases. There are like some of the literature-based text mining approaches we have developed for alternate animal models, and allergenetic predictive tools based on the genes, how it's going to impact this adverse outcome. That's also we have developed. And I'll quickly I switch the gears. In another 10 minutes, I'll wind complete this lecture. So right now, all the focus going on into developing drugs for potential drugs for to stop this pandemic, like targeting this SARS-CoV-2 proteins. I think KIET also did a good contribution, notable contribution in this domain of computational based efforts on developing some new drugs or identifying new compounds have potential to inhibit this SARS-CoV-2 targets. So this work, when we started like early during the early pandemic, like CSIR involved 5 verticals. One of the vertical is drug design. So we part of that vertical, we started working on it. So this is the time we created a snapshot, like how the scenario of drug development after COVID-19. As I said, like more than 3,45,000 of registered. Now it's huge number of new clinical studies got established during the time of COVID-19 pandemic. And we looked at the literature. There are several compounds, particularly we got a huge hit for chloroquine and hydroxychloroquine at the initial stages. And we start aware of these compounds also having some potential toxicity in terms of kidney or heart failure. So we thought of doing some analogs against this chloroquine and hydroxychloroquine using computational approaches. There are like more than 915 listed in WHO clinical trials, and many of them are right now in much more advanced stages. This kind of old word. So this is the kind of a word cloud identified like chloroquine and hydroxychloroquine that time is gaining much more importance. So we thought of taking those them and started doing a virtual screening against various structural protein and non-structural proteins. So this is our workflow. We identified from the zinc database 350 possible analogs for HCQ, like hydroxychloroquine, and 400 possible analogs for chloroquine. And this analog mining identified using various systematic processes like various ADMET and other docking-based scores. Finally, that docking-based scores, we started analyzing them using molecular dynamic simulations and how they going to interact, what are the compounds which can be more potential. And this study identified a final analog, which is non-toxic, very minimal level of um, toxicity, and some of them are which is still I cannot claim that, but still we need to do some experimental validation to see whether it's much more minimized toxicity level, and 5 analogs for chloroquine. So the first we generated the library, as I said, like chloroquine structure is shown in the center and hydroxychloroquine on your left. And we developed some analogs to it. And those analogs are screened for various ADMET properties and further screened into removed those compounds which are having this alert of various inhibition or adverse effect. So we removed them, and finally we got the selected compounds which are having potential to interact with the um, targets of this COVID-19 SARS-CoV-2 viral proteins, as well as we studied them with how it's going to interact with the host system also. So that's how we made a very comprehensive study. and we did the docking and followed by MD simulation. As I said, how this molecule at the time evaluation, it's go and interact and stabilize or destabilize the target or proteins. We studied this thorough study of 100 nanoseconds. Now it's showing only 50 nanoseconds. Further, we carried out additional 100 nanoseconds. And that resulted in one of the key identifying some of the compound which are having possible potential alternative to chloroquine and hydroxychloroquine. Further, we expanded study for various other repurposed drugs, and some of them are still under in press also, and some of them are communicated. So this resulted in paper in computational biology and medicine. It's got huge access during that particular quarter. And with that, like I would like to summarize this work. Like whatever I showed you is a glimpse of drug designing process, particularly highlighting computational toxicological perspective. There are a lot of advantage using in silico tools to predict the toxicity or properties to ensure the safety as well as minimize the in vivo or in vitro testing. That definitely going to impact both human health as well as environment and reduce, replace, and refine these 3R principles. And integrating this in vitro, in silico, and in vivo going to be strengthen this next generation assessment or development process. And we are providing solution much more reliable and realizable prediction across many domains, not only drug design, we are also working on safe product development, process development also. And our approach currently merging between toxicological as well as regulatory framework, how we can develop a good risk assessment platform that will enable the cost and time effective way for developing key stuff which is beneficial to the society. And I'd like to say that like now by a quote of Sidney Bernard, he told like all biology is becoming computational, much the same way it has become molecular, otherwise low input, high throughput, and no output science. And I'd like to thank my team members and the funding agencies. And I again, once again, thank the organizer for this great platform to share our work and enriching this kind of audience for this particularly virtual drug designing domain. And thank you so much.
Thank you so much, sir.
Thanks a lot, sir. Your lecture was highly informative, and I hope the participants would have definitely benefited a lot. This knowledge sharing was really useful for all the upcoming researchers. So now I request Ashumittal sir for his remarks.
Thank you, ma'am. Am I audible, ma'am?
Yes, sir.
Okay, okay. So really, ma'am, you rightly said it was a wonderful session, and we all are sure that we all participants enriched our knowledge in the appropriate use of in silico methods in drug discovery, development, chemical development, especially associated with the toxicity prediction and toxicity profiling. So I would like to share some learning takeaways from this session. What I understood from this session, that in silico methods, we can develop these methods for or we can apply these methods to drug designing and hypothesis development and testing related to toxicity predictions while developing the chemicals and drugs. And these in silico computational approaches, they include quantitative structural active relationships, similarity searchings, and docking and molecular modeling simulations, the use of machine learning data mining databases. Also, sir discussed about network analysis tools and data analysis tools that computer can use to profile the toxicity and for safety assessment. Now, such methods, what we understood that such methods are frequently and applied in drug discovery, chemical development, optimization of lead molecules, identification of potential targets for incurable and preventable diseases. Nowadays, it is used for development of vaccines. So so we thank Dr. Parthasarathi for such an enlightening lecture. on appropriate use of in silico methods in drug development, chemical development, especially associated with toxicity profiling and safety assessment. So we thank you, sir, for such wonderful lecture. And I request the participants to kindly ask their questions.
Thank you.
Vishali ma'am, can you see some hands? Okay, sir, I have one question. May I ask?
Please, sir, please.
Sir, when to apply this Lipinski filter, before docking studies or after? Because it may leave out some potential compounds. So what is your opinion, sir?
Actually, that is when just I'll give you first general perspective, and then I'll tell you about what we are currently doing. General people are using in, you are pointed very correctly, they are using in both way, before as well as after they are using it. Like Lipinski taking the bunch collection of chemicals, then people started applying Lipinski rule of 5, then further taking for docking. And in other trends, what I'm saying it, people are started using huge number of screening with small molecular dynamic simulation. Also, people started doing, including docking. Then started finally come up with Lipinski rule of 5. These are the two procedure people are following it. And what I prefer is like, first, I want to create a huge data set, and I want to go complete screening. Whether if it's not even coming into the Lipinski rule of 5, I don't want to take and put my efforts on computational resource on molecular dynamic simulation, docking based screening. So first I will do with Lipinski rule of 5 of entire collection of the data sets, and then I'll go for further docking or virtual screening process.
Okay, thank you, sir. So there's one more question, sir. So many freely available linking tools are available on the website. So are they efficient and reliable to link different ligands in silico drug designing, sir? Can we rely on those freely available tools?
Yes, some of them are kind of very that they developed in much more rigorous way. They are mostly reliable, and some of them are not that so rigorous in terms of data involved in it. So you need to be a little more critical on it. Like that's why we did some kind of benchmarking analysis first, and known molecules you can test and see whether they are predicting in a better way or not falling under the scope of your study. Then you can choose these online tools which are available. And some of them are for some basic screening and all. Okay, you can start with it. You can start using this online screening for learning purpose. When you are starting working on R&D, definitely you need to have a critical questions like whether it's valid and how much it's kind of reliable on the system. So you have to do some kind of small screening or benchmarking analysis to see the validity. Then you can go ahead and start doing your R&D. For learning purpose, just go ahead and try some of your curiosity on doing your science on drug designing.
Okay, thank you, sir. So can we use this in silico based approaches for developing the natural products also?
Yes, yes. That's like actually there are several efforts, several research group currently working on small natural molecules or natural compounds, bioactive compounds, using generating in silico approach, an in silico based predictive properties, and started building databases and started comparing with already existing efforts and efficiency of those compounds. And you can definitely use this new in silico approaches for natural product design and development. That's having huge advantage currently. Like actually, as a like we can also do some safe by designing approaches. Now you can also fine tune the groups or fine tune or add the groups or identifying which group is really targeted, and how our traditional knowledge also built upon like combination therapies. So that's also people are currently working on using computational approaches. Yes.
Okay, thank you, sir, for
Hello.
answering our questions.
So
So
there
is there,
are some questions from audience.
Good afternoon,
You are
sir.
allowed.
Good afternoon.
Yes, Vinay sir.
Sir, myself is Dr. Vinay. I'm in KIET School of Pharmacy. Sir, actually, it was very nice presentation and very useful information that you have provided. Actually, I have experience on working on this software in silico toxicology. So I just want to know that as there are number of databases or software are available like Direct Nexus, Topcat, Toxtree. So how do we select which one is the best one that we can use for the toxicity assessment, especially genotoxicity?
Okay. Yeah, that's a great question. So for computationally, we do everything. Yeah, I use both Toxtree. I use Seranexus and like Derex and Seranexus, Topcat, and depends on which property. Sometimes I compare this ADMED properties from various tools also, like Topcat, along with some other online tools, some of the most reliable sources, and also based on literature, we also go ahead and critically review the literatures, which tools or which properties or which series of system we are going to choose in the study, and what it's already people are working in that research R&D domain, and some of them are having advantages, some of them are must be discrepancies are found in that utilizing these in silico models. Based on that, we do some kind of proper level of screening first, then only we choose the model. And some of our exploratory projects, what we do is like okay, the system is not much relevant or it's not fall into that categories, so we started expanding our scope in trying to use both expert based and knowledge based research of various tools, and how they we develop a consensus based system, like what and which one having a right combination or it's not a right, it's going some kind of a adverse effect it's showing it, and how maybe if you call them like 10 compounds, if it's predicting 9 or right, only one is wrong, or 7, then 3 is wrong and 7 is right. Based on that, we set an kind of a I'm not statistically speaking, I'm just numbering, I'm speaking. So based on that, I'll select the particular model for our study. And mostly right now we have started using screening of like couple of tools and see which one is working better, then only we are going ahead for our further studies.
Thank you, sir.
And
Actually,
depends on the problem you are choosing, like toxicity endpoints versus genotoxicity. Also, like very few are available. That's how I shown you in Vega platform. What are the things are available? Sorry, excuse me.
yeah, sir.
Yes, Miss Devlina Tiglani.
Yes, ma'am. Sir, I have two
Just wait.
questions regarding this. I
Pardon?
have two questions regarding this topic. Actually,
Yeah,
I
yeah,
am
please.
trying to learn simulation, and there is a problem
Uh-huh.
in the script. So, do you have any suggestion regarding that to tackle these? Because I have tried multiple, like you know that Google shows various of options, and we are not sure which one is the correct one, which one is not. So, how do we know that which one we have to use for the correct simulations to get right answers or the right docking results for that? And my second
Uh,
question is that, like we do some toxicity testing virtually and docking studies. In various studies, I've seen that there are some compounds which are showing highly toxicity in virtually, while they have shown the best result in the docking. So, how do we compare these results to achieve or to get the best compound that we dock?
yeah, there are two problem, Sushil, with that. Like you are asking. Yeah, the scripting is very important. Like that, you probably you need to learn which algorithm you can you are trying to utilize it. So that is more or like your interest. Right now, probably whatever the automated tools are available, that that's like kind of for me. That's I'm telling that like all my that's become very lazy to our researchers. So you have to be strengthened upon like all the components of this kind of a computational evaluation process. So algorithm and the programming are like kind of a small scripts. that need lot of time and lot of like kind of learning process. So how you can do the bug fixing. Sometimes like it's more like kind of not much your chemical, physical, biological understanding. It's more like your mathematical and analytical skills, how you are going to develop upon. That is what is very important for your algorithm development and finding your errors in your like kind of your scripts. So that will easily kind of streamline in terms of what kind of a component you are going to script upon. Like how right now the automated script also we have developing in our laboratory. Like we can do like screening, not only using the multiple methods or multiple components. If you choose selected structures, and it will go and search in the databases. That databases, once you got that series of molecules, that can utilized for various screening including like softwares. You are talking about Topcat or which like Dera, Sara, Nexus. It can automatically go and fetch the particular toxicity in points like that. I need to give an input, so that will fetch, and then it will do a statistical analysis, and finally the selected molecules can go for toxicity like kind of a docking screening. So yes, if you have the problem with highest binding scores or docking scores, but it's going to be a toxic, definitely you need to verify and research which are non-toxic. That's what I can suggest you. But which group is going to be toxic, if you identify it, just that will give you an ample amount of another research direction for you to replace that particular group and start doing your R&D work. Okay. Sorry.
I hope, Devlina, your question is answered.
Yes, ma'am. Thank you, sir.
Okay, thank you. Please unmute Mr. Nishan Kumar. He has raised his hand.
Sure. Yeah. 2 minutes. I have to go for another
Ma'am,
meeting.
they can unmute themselves now. I have
Okay,
removed the
sir.
restriction. They are already unmuted, ma'am.
Yeah.
So this is the last question. If any other are there, we will ask them to connect with you through
Yeah.
mail.
Please connect with me offline. Definitely, I will happy to answer. 1:30. I have yeah our
Yes, sir.
director some kind of quick meeting.
Right, sir. Right, sir. So, dear participants, you please write up your questions. I will share sir's ID, or you can write us to us. We will share it with him. Dipti, ma'am.
Yes, ma'am. So, sir, just 1 minute more, sir. Sir, I request now Ashu Mittal sir to please present the certificate of gratitude to Dr. Parthasarathi as a token of remembrance of the event. Ashu, sir.
Yeah, thank you, ma'am. So, yeah. So, ma'am, sir, I present you this.
So the screen is getting shared. Just a minute.
It's a virtual presentation as a
Thank you
token of
so much.
affection. Please accept, sir. This certificate.
Yes.
So we
Thank
look
you so much.
forward
So
some
nice
more
of you.
collaborative efforts or some more collaborative projects with you. Thank you so much, sir, and thank you very much for your valuable, precious time.
Yeah. Thank you. Thank you.
Thank you,
Yeah.
sir.
Thanks for the opportunity.
Thank
It's
you.
great interacting with you all. Definitely, we can strengthen up this process in the course in the coming years. Thank you.
Yes, sir. Thank you again, sir.
Thank you.
Thank you, sir, and thank you, everyone. Thank you, participants. So now we break for lunch here, and we'll meet at 2:30 for the technical session 2, day 1. So now we'll
And
meet at 2:30.
ma'am, we are enabling the chat box also. So if you have any questions, you please post those in the chat box so that those will be taken on priority, or we will unmute you. Thank you so much, everyone. Please enjoy
Just a
your
moment,
lunch.
just a second, ma'am.
Sir. Sir,
Uh,
please.
the link for attendance has been shared in the shared via WhatsApp, and if anyone is left from filling up the attendance, they may do it by 1:45. After 1:45, the link will be disabled. Thank you so much.
सर आप में से कोई यहाँ पे है अभी टीम्स पे कोई लॉगिन रहेगा?
Hello.
सूर्या सर?
Ma'am, I am available, no problem. Vishali ma'am, I am there.
ओके सर, एक्चुअली मैं डीन ऑफिस होकर आ रही हूँ सर आधे घंटे में ना, तो प्लीज
मैं हूँ
आप कोई भी
लॉगिन
एक
मैडम,
I am here, ma'am. No problem.
मैं
ओके
लॉगिन
ओके,
हूँ
थैंक यू सर।
ऑर्गेनाइजर, तो आपको कोई प्रॉब्लम नहीं
ओके
होगी।
सर, थैंक यू, थैंक यू सर।
Hello.
Yes,
Kindly display the lunch picture. Vidhu ma'am, can you just I have made a PPT for virtual lunch. You just share that.
sir, have you shared it in the WhatsApp group?
Yes, yes.
I'll just display.
विधु मैम है अभी। विधु मैम, आप एज अटेंडी ज्वाइन करा है, अपने टीम मेंबर ज्वाइन करा हुआ है?
Sir,
टीम मेंबर है, टीम मेंबर।
आप टीम मेंबर है?
I'm just sharing it, sir.
नहीं नहीं, विधु मैम कोई है आपके पास जो अटेंडी की तरह जिन्होंने ज्वाइन करा हो?
हाँ, किरण मैम है, अभिषेक सर हैं, अटेंडी लिस्ट में दिख रहे हैं।
ओके,
अवधेश सर
अभिषेक
हैं।
सर, अभिषेक सर आपको आवाज आ रही है मेरी? वो सर अटेंडेंस कितने लिंक मत पोस्ट करिए सर।
Himanshu sir, recording is on actually. इसको ऑफ कर दीजिए पहले. Whatever we are discussing here, ना, it will go on recording.
ओके, प्रवीण सर अभी ऑफ हो गया ना?
Just a minute, sir. No, sir. Recording is started yet.